Welcome to the Patterns of Inheritance lecture. I just want to make a note for this lecture moving forward. We're going to talk about sex chromosomes, the X and Y chromosome, the X carrying genetics for becoming female, the Y carrying genetics for becoming male, and we'll talk about those chromosome combinations of XX and XY from female and male. Yes, there are patterns in which there are created genetic variations that are between those two extremes on a spectrum. We'll talk about that as well. The one thing to remember here, though, is that when we talk about phenotype, phenotype is a direct relationship to genotype in this lecture. Phenotype in this lecture does not include the concept of gender identity and the spectrum along which a physical appearance can exist for gender. So I just wanted to make that clear that this lecture very simply talks about the chromosome setup of XX, XY, and variations between, and the direct relationship to physical appearance without amendments. Back in the 19th century, an Austrian monk named Gregor Mendel, he had a lot of time on his hands. And so he decided how to grow pea plants, like the ones shown here. He wanted to determine inheritance of different traits. And so he investigated how individual genes were inherited by breeding these plants and making very careful observations on the parents and the offspring. He bred hundreds of generations of these plants. And he noticed patterns of inheritance that he described in his two laws, the law of segregation and the law of independent assortment. Gregor Mendel's work wasn't really appreciated until about 50 years after he published it. I don't think that his obscurity was due to the difficulty of his research. Instead, I think that many biologists just weren't ready for Mendel's work, at least not until they understood chromosomes and the role that they play during meiosis and fertilization. And that understanding did not come until the early 20th century. Let's set up some terminology because I love the vocab, and this will become very important for answering questions on genetics. When students have a misunderstanding of those Punnett squares, I find it usually comes from the misunderstanding of a vocab term. The question is asking you this, but you think it's asking you that because you don't really know the definition of one of the words in the question. Okay, so you and I are both humans, which means that we both have the same genes. And you might be thinking, Amy, you and I definitely do not have the same genes because we are not identical twins. Yes. But the word gene just means location on a chromosome. On my third chromosome, at a certain location, I have a gene for making me, 2 arms, let's say. You also have that gene at that same place. In fact, every human does. That's what makes us human, partly. But your gene says to make long arms, and my gene says to make short, stubby arms. There are different versions of genes, and these versions are called alleles. So Mendel looked at the gene for plant height, and there were 2 variations of that gene, or 2 alleles, one for making a tall plant and one for making a dwarf plant. In this class, if you go to use the word gene, you probably should be using the word allele. Get used to it. You may recall that we established in an earlier module that your chromosomes they're like socks in that they come in pairs. Instead of left and right, you have a chromosome from mom and one from dad. So this means that for any one gene, for any one location on a chromosome, you would actually have 2 DNA sequences for that gene, not one, as shown in the picture. The combination of alleles for any one gene is called your genotype. And here, we seem to have two alleles that are different. But obviously, you could have two alleles that are the same. Either the green piece is the same of DNA or the orange pieces, either one. But there's also a word that we need to get to know, and that's phenotype. This is their phenotype, what they look like. It's a little bit more than your physical appearance, like a widow's peak or freckles are your phenotype. Your phenotype, it can also be your blood type or your propensity for cardiovascular disease and much more. A student once told me that they remembered phenotype because phenotype and physical appearance both begin with PH. But you have to remember it could be a little bit more than physical appearance. If your trait is genetic or determined by your genes, you have a genotype or DNA on your chromosomes that will determine the phenotype. For these plants here, we have two phenotypes or appearances that are possible based on the alleles that exist in the plant population, tall plants or dwarf plants. So we get the sense that genotype determines phenotype. Yes, it does. Although Mendel couldn't have possibly known it at the time, he was describing the behavior of homologous pairs of chromosomes during meiosis and fertilization. Remember, chromosomes are long pieces of DNA, each one typically containing hundreds of genes. And humans have 46 chromosomes. Pea plants have 14. These chromosomes, they come in matched sets called homologous pairs. And humans have 23 homologous pairs of chromosomes, and pea plants have 7 homologous pairs. You have to think of homologous pairs like socks. No one ever says, I have 46 socks. All right, well, my husband might, because he's one of those people that buys, like, an industrial-sized pack of socks that are all the same. I, on the other hand, I have pairs of socks that feel different, and thus I will not mix and match from the pairs. So I would say that I have 23 pairs of socks. And this is how you refer to your chromosomes, 23 pairs. The pairs are like insurance. If one chromosome doesn't work, hey, no problem, you have the other one. It's like that with all the important organs in your body, kidneys, eyes, lungs. One goes bad and the other one is like, don't worry, I got this. So don't forget this. Mendel didn't have the benefit of knowing about chromosomes and different sequences and different alleles. You do. I really do love this picture here. I took it from an animation that is sadly no longer on the interwebs. This illustrates the law of segregation. Let's look at mom and dad there. Next to them is a picture of their 46 chromosomes paired up into their homologous chromosomes. And we can see that dad has 23 pairs, and we can assume that the green chromosomes came from one parent and the red ones came from another. Mom has 23 chromosomes as well, 23 pairs. One set blue from one parent and the other one yellow from the other parent. When they go to reproduce, the father donates one of each pair. And whether it's green or red, that's only a 50-50 chance. Same with mom. Note the combination of chromosomes. Take a moment right now and really look at this picture. Note the combination of chromosomes and the colors of them for the baby. I feel like this picture explains it in a way that words just can't. Take a moment. I'm taking a moment too. All right. Mendel, he proposed that for each characteristic he studied, that a pea plant had 2 alleles, or alternate forms of a gene, one obtained from the plant's mother and one from the plant's father. And Mendel's law of segregation states that these 2 alleles separate from each other when gametes, sperm or egg, are produced. That is, each sperm or egg gets only one allele of a given pair. In short, Dad could give both green or red, but not both. What is the probability of giving green or red? 50-50. 50-50. Now let's talk about Mendel's law of independent assortment. Mendel stated that when more than one trait was considered during a genetic cross, like the inheritance of each trait was independent of the other traits. For example, whether a pea plant had purple or white flowers had nothing to do with whether it was short or tall. Flower color and height were inherited independently. In fact, he found this relationship held for all 7 traits that he studied. And he was lucky. That's not the way it works sometimes. So to put it in terms of chromosomes, each pair of alleles segregates independently of other pairs during gamete formation. And referring to this picture again, Dad will give green at chromosome 1, and that has no bearing whatsoever on whether or not he gives green at chromosome 2. This is like tossing 2 coins at once. One coin has a different allele for flower color on each side, and the second coin has a different allele for height on each side. Every time a gamete is formed by meiosis, the 2 coins are flipped, and the outcome determines the genotype of the gamete. Neither coin affects each other, so any combination of heads and tails is equally likely. This is the case for traits of yours such as freckles and a widow's peak. Whether you get freckles has nothing to do with whether you get a widow's peak. The law of segregation applies to both Mendel's genetics and some more complex genetic relationships. Mendel's principle of independent assortment, it works as long as you're referring to genes that are on separate chromosomes. And Mendel developed his theories concerning inheritance by observing genes that have only 2 alleles, and that these alleles have a dominant recessive relationship. So this accounts for some easily observed traits in humans, such as having a widow's peak or not, having freckles or not, having detached or attached earlobes. Phenotypes can be dominant or recessive. How do you know? How do you know if freckles is dominant? I mean, I know that you can tell if you have freckles or not, but how do you know if freckles is dominant or recessive? You don't know. You're told. The dominant-recessive relationship of genetics, it's predetermined, and you will always be told which is dominant and which is recessive. Questions on the lab will set you up with this information. It will be your job to get this info from the word problems. One more thing, dominant doesn't always mean more common or more popular. In fact, polydactyly, or having 6 digits on a hand or a foot, is a dominant trait. However, it is not more common. To be prepared to answer questions on genetics, understanding terminology is very important. To begin, these are chromosomes. The purple and white bands are the locations on these chromosomes for flower color. Obviously, you have 2 chromosomes for each individual, and that these chromosomes, they have these genes. So you have a genotype, which is your combination of types of alleles. If you have 2 dominant alleles, you are called homozygous dominant. If you have 2 recessive alleles, you are called homozygous recessive. If you have 1 dominant and 1 recessive, you are called just heterozygous. These are the terms that we use when describing an individual. If you are ever asked for a genotype on a question, these 3 terms are how you answer. All right, so what does this mean for what you look like or for the phenotype? That has to do with something that Mendel observed from breeding his pea plants a gabillion times. He noted that there was a specific relationship between the dominant and recessive allele. Whenever a dominant allele was present, it was expressed, no matter what the second allele was. He also noted that the recessive phenotype, it was only expressed when two recessive alleles are present. These observations of Mendel's give us some rules to follow. The homozygous dominant and heterozygote have the dominant phenotype. The homozygous recessive has the recessive phenotype. Again, how do you know which phenotype is dominant or recessive? You're told that. Let's use some letters for a little shorthand terminology. Let's use the letter A. In traditional genetics notation, we designate the dominant allele with a capital letter, in this case, capital A, and the recessive allele as the lowercase letter, in this case, lowercase a. Let's apply this to flower color. A purple flower color, or phenotype, occurs whenever the dominant allele, or capital A, is present. A white flower color only occurs when two recessive alleles, or two lowercase a's, are present. A plant with purple flowers could be homozygous dominant, or it could be heterozygous. A plant with white flowers can only be homozygous recessive. But how do we determine if a plant is homozygous dominant or heterozygous? Well, you need a little bit more information about either the generation before or the generation after. We would also need to apply Mendel's law of segregation to those two generations. And we'd need to use the logic of something that is called a Punnett square. Punnett squares. I have another PowerPoint in which I work a bunch of Punnett squares. In this slide, I'm just going to define them. So the first thing to know is that Punnett squares show the probability of offspring. They don't show you what you get. They show you what you can get, what you might get, possibilities. And then probabilities can be calculated. They will be calculated in the lab. We know that Punnett squares usually have a lot of letters in them, right? And they kind of look like this picture down here on the bottom. A little complicated. Can you see the words sperm and egg along the left side and the top side of the square? These are the gametes produced by each parent, along the side, along the top. The Punnett square takes these gametes and it combines them, just as in fertilization. Sperm plus egg equals zygote. Therefore, each square of the Punnett square shows you a possible zygote that could develop. Each square answers, what kind of baby can I have? And then we can calculate the probabilities or percentages as to how likely a certain type of baby would be. Remember, I have an entirely different PowerPoint for working these different types of Punnett squares. Let's recap before we end. These are the rules for Mendel's patterns of inheritance. I just want you to kind of like read them and review them very quickly here as a reminder. These are things that you really want to keep in mind as I cover what's on the next slide, which is about exceptions to this rule, right? There's always exceptions. Okay, some genes are located near each other on the same chromosome, a situation called gene linkage. Linkage is actually a big exception to Mendel's law of independent assortment, since the genes can be inherited together because they are on the same chromosome. They're not independent. We see this with pigmentation traits in humans. We have some common pigmentation schemes like the light complexioned, light hair, blue eyes of the Swedish, and dark pigmentation, dark eyes, and dark hair of African populations. Yes, genes for pigmentation can be inherited together. But crossing over in meiosis 1 can break that linkage between genes. Remember, crossing over was that exchange of DNA between the homologous chromosomes in meiosis 1. This exchange will happen for the one chromosome carrying pigmentation genes, exchanging maybe one or maybe a few of the pigmentation genes. Like gene linkage, nearly all of the exceptions that geneticists have found to Mendel's basic laws are due to the behavior of chromosomes. One very interesting set of exceptions that you could explore further on your own is sex-linked traits. In such cases, genes located on the sex chromosomes or on the XY chromosomes, these chromosomes follow usual patterns of inheritance, right? They're passed down in meiosis and fertilization like all of your other chromosomes. But the relationship between the X and Y chromosome is just a little bit different than the dominant recessive relationship. Think about the shape of the X and Y chromosomes for a minute. The X has a lot more DNA in it than the Y. There are genes on the X that are not on the Y, like genes for hemophilia, a blood disease, or genes for color blindness. Therefore, the XX combination of females produces 2 chances to get a gene that gives you normal color sight and not be colorblind. But with the XY combination of men, they only have 1 chance to get the normal color sight allele. This is why there are more men who are colorblind than women. It's just the math of probability. So I want you to remember that at first, such cases like this, they seemed like exceptions to Mendel's law. Oh, they didn't follow it. But further investigation kind of revealed that the chromosomal basis of inheritance, what happens in meiosis, can explain these exceptions as following Mendel's law, but being interpreted a little bit differently.